And thanks to everyone for joining us for this month's free Orion webinar titled Seeking Best Practices in Classifier Construction and Testing. I'm pleased to introduce Clark D. Jeffries, a bioinformatics scientist with the Renaissance Computing Organization of the University of North Carolina at Chapel Hill. His interests include interpretation of neuroscientific information, especially that pertaining to schizophrenia. He was previously a professor of mathematical sciences at Clemson University, where he was an author on more than 50 scientific papers. Following that, Dr. Jeffries was a designer with the Microelectronics division of IBM, where he was an inventor on 117 issued U.S. patents. Now, he may not know this, but during his PhD studies at the University of Toronto, Dr. Jeffries was the reserve kick holder for the Toronto Argonauts Canadian Football League franchise, where he set a record of 17 successful field goal holds in the preseason, according to Dr. Jeffries' Facebook page. Unfortunately, neither the team nor the CFL tracked this statistic. Fun facts to know and tell. And before his academic career, Dr. Jeffries was considered one of the foremost lyricists in popular music. His brief career was highlighted by a still record 12 Grammy nominations in 1 year. Unfortunately, he lost in every category and never wrote another song again. But music's loss is science's gain. Thank you for joining us. Sit back and enjoy Dr. Clark Jeffries, Seeking Best Practices in Classifier Construction and Testing. Over to you.
Okay. Well, thank you very much. I can say I've never had an introduction like that before. Thanks very much, Lenny. Yes, the talk will be a combination of my own opinions and also some facts that might or might not be well known about building classifiers. The impetus for this is widely known and appreciated, and here is just one of the papers that's appeared on the topic, and it was written by Francis Collins and his colleague, Dr. Tabak at NIH. And of course, those of us who are involved with NIH research, we pay attention when Dr. Collins writes an article, and this one was, well, a pretty pointed article. He touched on the topic of reproducibility and why so much costly research published in high-quality journals doesn't turn out to be very reproducible. Well, there are three facets of this problem of reproducibility. First is consistent definitions of case control and selection of the subjects, which in itself is a difficult problem. The scientists and medical doctors and clinicians who engage in this recruitment of subjects and so on, this is a very complicated and demanding task. And if it's not done correctly, well, everything else fails. For example, and this is touching on topic 2 here, it can happen that blood samples or other samples deteriorate quickly after they're collected, and in some substances may be misleadingly high or low in the subsequent assays. So that's the sort of thing that the experts who draw the samples and collect the tissues or do the interviews or whatever other types of assays there are, they must be constantly aware and sensitive to those problems. Well, so then the third part, and the part that I'm concerned with, is the consistency of classifier construction and testing. So even if the definitions of case control and the selection of subjects and the lab assays are all done properly and consistently, there's still the problem of going from numbers to conclusions. And the situation is that modern classification generally depends upon is multiplex. It depends on a combination of assayed parameters. And it can happen that the combination does one thing, but individual parameters may not, in general, follow that trend. And so it's building this combination that's the hard part. And that's the part I would like to focus on. Now, what can go wrong? Well, a few years ago, as I'm sure many who are listening know, the Nature family of journals published guidelines for authors that included the following statements. The statistical tests must be properly conducted. And many statistical tests require normal distributions, or what is called here approximately normal distributions. And when using these tests, the authors have to explain how they tested it for normality and so on. Furthermore, if there is a small sample size, some statistical tests are not appropriate. And both of these topics have been roundly ignored in the scientific and medical literature. And this has been a big factor in the failure of reproducibility. But given these, I'd like to just go through an example of what can go wrong. And it's the sort of thing that everyone who's involved in classifier construction should know about and be sensitive to. Here is some made-up data just to illustrate a point. So there are 10 cases and 20 controls. There's nothing really extraordinary about these, I suppose. It happens that the student t-test p-value is 0.04, which is below the customary level of 0.05 for significance. Now here is an example too, pretty similar, but this time the same test yields a p-value of 0.25, which is quite a bit above 0.05, and therefore one would ordinarily or customarily dismiss this test as this example as being not statistically significant. Well, here are the two together, and I think most persons looking at them would say there's really not a great deal of difference. So the first 10, in fact, the cases, they're identical. They are exactly identical, and I've just played around with the controls to alter the p-value from 0.04 to 0.25. Now, as it says in the Nature guidelines, this test depends upon the assumption of approximate normality, and there are two tests for normality that are commonly used, and here is one of them applied to the cases. It's called the Kolmogorov-Smirnov test, and this is from a commercial program called Unscrambler, which I like a lot. And I'll just say that without going into details, this program applied to the case data that I just showed you does assure us of normality. Now, technically, what it assures us of is not normal. There is a logical difference there, which statisticians appreciate. Here is the same test applied to one of the two sets of control data, and here's the same test applied to the other set. And the upshot of the last three slides is that they do pass conventional tests for normality, to speak approximately, fairly well. So there's no funny business in these examples. The problem is the experimenter must look at the raw data and think long and hard about what is really going on and whether these are really significantly different, and not just blithely apply a customary test and then write this into a paper and publish it. In other words, there are a lot of things that can go wrong, and the prudent experimenter and the prudent analyst of the data must be aware of them. Now, an alternative method, as it says in the Nature guidelines, is non-parametric analysis. Non-parametric analysis, for the most part, means assigning ranks to data. So one throws away the details of the raw data and instead arrives at integers. And so the benefit of doing so, throwing away that information, is that we no longer need to pay much attention to this onerous task of proving normality. And so these are 20 real numbers, 20 representations of real numbers, and there are 20 ranks. And this approach enables us sometimes to get away with very few cases and very few controls. And here is an instance where there were only 3 cases and 3 controls, in which I dealt with 3 years ago, and which illustrates this point. Now, Suppose we have 3 cases, 3 controls, and 100 markers. So the analyst would say, well, this is a 100 by 6 matrix. And we have 3 cases, 3 controls. And for each marker, we can partition the 6 readings, or the 6 values, into 3 high and 3 low. 3 high values, 3 low values. And so here are the very first line there is case 1, case 2, case 3, all greater than control 1, control 2, control 3. If one thinks about it for a moment, one realizes there are 20 different ways to do this. Now, with 3 cases and 3 controls, 6, there are actually 6 factorial, or 720 different rankings. But for the sake of this illustration, we only think of the 3 high and the 3 low. And a moment's reflection reveals there are 20 such rankings. And as a consequence, if everything inside is random, then of the 20 different types of rankings, we would, and 100 markers, we would expect 5 markers on average. And well, the expected number of markers in each of these 20 types would be 5. So with random data, this is what we would expect. Now at the 2 extremes, the very left and the very right, we have all 3 cases greater than all 3 controls, or all 3 controls greater than all 3 cases. And those, of course, are the most interesting, because those tell us that those markers that showed up in those bars of this chart, those markers were distinguished by being consistently higher in cases than in controls, or vice versa. The 18 other possible combinations in the middle aren't so interesting. And here is another draw of random values, and this is what they look like. Now, if we do this 100 times and count the maximum number of markers in each of the 20 categories that we ever see, well, when I did it, I saw the maximum was 13, and it was attained 5 times. Okay, so these are the data that one would expect. This is the null result, where the what's going in is purely random. Now these are the real data, and notice the great contrast at the 2 ends. In the real data, there were 21 of the markers which had 3 cases greater than the 3 controls, and there were 31 where the 3 controls greater than 3 cases. Now this is very good news, because it tells us that far beyond reasonable expectation, there are many of these markers that are informative of the experiment. And all the details on this experiment appeared in the journal RNA a few years ago. And so this was a real experiment where we found, using only 3 cases and 3 controls, the 3 cases actually were nuclear fractions versus cytoplasmic fractions from human neural progenitor cells, but that's not really crucial to the argument. We found, with only 3 cases and 3 controls, such a strong difference that it could not reasonably be ascribed to chance. So the point of everything I've said so far is routine or simple application of statistical methods can lead to a lot of trouble and account for a lot of failures of reproducibility. On the other hand, thinking things through and trying a lot of random, trying by brute force a lot of random simulations can lead to some results that are pretty clearly not by chance and very likely to be reproduced. So in the background notes here, I would say we have X cases and X controls. So this is leading to a kind of general methodology or best practices for building classifiers. The basic steps are to normalize the data, of course, and then to build a classifier, of course, and to evaluate a figure of merit. Now, the normalization method, the construction of the classifier, and the figure of merit, those are all user's choice. A great deal has been written about these different methods, and of course there are proponents of one method or another. But in a way, the ultimate test of success does not depend on them. And those who would want to follow up on this topic, I would strongly recommend having a look at this paper by David Hand. He's a professor at London Imperial College London. He's a past president of the Royal Society or Royal Statistical Society. Who, he's in a long chain of very prominent statisticians. In the 50s, the president was R.A. Fisher or Sir Ronald. So, this method, this paper generated a lot of friction and a lot of excitement in the statistics community because it basically said it doesn't matter what methods you use. And of course, this is stepping on the toes of those who invent different methods and want to put them forward as the best answer. But I think this is a great paper, and in fact, it's and a great issue of this journal, where there's a lot of debate about choice of classifier methods. At any rate, then this best practices approach demands us to do the following. We randomly permute the labels of all the X cases and Y controls. So now we have pseudo classifiers constructed from pseudo data. And the, I guess best practice would demand at least 100 times and preferably at least 1,000 times that one go through this tedious exercise. The crucial points, of course, are that the normalization, the classifier construction, and the selection and evaluation of figure of merit and the tabulation of all these results, all of these things have to be exactly the same. So if your classifier used at some stage a pair of dice, well, you should try to use the same pair of dice. Now I'm being a little silly, but it's crucial that the raw data be permuted in terms of X case, Y control labels and otherwise be treated exactly the same as the true data that led to the true classifier. So then we compare the merit of the true classifier applied to true data with all the pseudo classifiers applied to their respective pseudo data. And when the true classifier outperforms most of the pseudo classifiers, well, that gives us some evidence of the credibility of the true classifier. Now, a problem is that random labeling tests can be computationally expensive. So this implies to the model builder that it's a good idea to select normalization and classifier algorithms from the beginning with computational complexity in mind. Because one way or another, all of these steps, all the steps that the person selects must be repeated many times to conform with random labeling testing. Then, if the random labeling applied to all the data at once, if that works to the satisfaction of the modeler, then it's a good practice to go back and okay, now you've settled on a way to build, to normalize and to build a classifier. It's the next part of the good practice, or next good practice, is to go back and apply this classifier construction method to many subsets of the original full data, 80% subsets of cases, 80% subsets of controls. And then, and this is the part that's the mystery still, somehow integrate all those classifiers into one. Well, of course, this depends on the choice of classifier. And if I know how to do it with a restricted set of classifier types, but how it's done in general, well, that rests with the classifier builder. So then we compare the outcomes for doing this of true data with on true data. We do this 1,000 times with the pseudo data, and if everything works, if all of these processes point to a good performance of the classifier, then we have an indication of three important statements. First of all, the raw data actually contain information about case and control. Of course, it could happen that they don't, and that in itself may be an important finding. Second, the normalization process must not have destroyed the information, right? If you have these differences and they come up after all these random labeling and 80% subset tests, well then it cannot be true that the normalization process, whatever it was, destroyed the information. And finally, the classifier construction partly discovered the information. Now, it must be added, however, as shown here, all of the above certainly, these exercises certainly do not imply that the classifier is unique or optimal. It could well be that someone else with some other take on classifier or normalization will come up with a system which is better. One never knows that, really. One can only say that it's, at the end of the day, that having passed all these tests, that the classifier constructed was a good one, not the greatest. Now, the really big problem is, in my opinion, is that there are many simplified methods, simplifications of the stringent random labeling test. And these simplified methods, in certain circumstances, can be deceptive, and they can lead to failure to reproduce findings. Now, this is an example of what random labeling testing should look like. So this is a classifier method applied to all the data and 1,000 times, and it generates a histogram. Now, in the histogram, the orange rectangle here, of course, represents that the AUCs, the area under curve performance for some of the classifiers in these random trials, random relabeling of case and control, 68 of them had values, the AUC's values were in this 0.73 to 0.76 bracket. And we also see, though, that the true classifier, the black dot over to the right, had an AUC of 0.884. Well, this is happy news. But this graph is indicative of a couple of very important points. First of all, it's sometimes said that a random classifier will have an AUC of 0.5. Now, that applies to a random classifier means that it simply guesses case or control in light of the known proportions of cases and controls that are to be examined. It also implies a great number of cases and controls, at least more than 10 and 10, something like 50 and 50 or more. It also implies that the underlying data do not have extensive hidden interdependencies. Now, real biological data have plenty of interdependencies, and this particular graph was taken from a study of microRNAs, and microRNAs, for those who are familiar with that topic, microRNAs are highly interdependent. They're regulators. They have very similar, some have very similar sequences. Some are thought to be nearly redundant. Some on chromosomes are physically transcribed at the same time because they're so physically close. So, and this is an ocean, really, of interdependencies, and it cannot be fathomed. So the only thing one can do is to simulate it. And in this simulation, we saw that there were many cases where mixing up the cases and controls, and by the way, there were 31 cases and 37 controls, mixing them up in this random manner still gave us fairly nice AUCs, 0.8, 0.85 even. So calling it a day done by achieving an AUC of 0.8 or even 0.85 is not always a good idea. It can be that was achieved by chance. And another point on this graph is that we see that there are a few times when mixing or choosing these, from the mix of 68 samples, choosing 31 controls and 31 cases and 37 controls. By chance, that choice was such that the classifier method actually produced, from this nonsense data, it produced a classifier that outperformed the true data. So that can happen too. Now, happily, in this particular instance, this distribution, which is a beta distribution, gave us a p-value that was slightly less than 0.05. And I myself, I don't really ascribe a great deal of meaning to 0.05 versus 0.04 or 0.06 or even 0.1 and 0.02. But the point is rather that the AUC was pretty respectable, 884, and it appears to be significantly higher than most of the random labeling tests that were illustrated here. So to recapitulate, there were 1,000 trials here. We randomly permuted all the case control labels of all the samples and repeatedly applied a classifier algorithm, which is, as I said, that's user's choice. I made a particular choice and applied it. This is what happened. We tabulate the AUCs, and this is the kind of graph that you should see. Then we integrate multiple 80% classifiers. In other words, we take 1,000 times 80% of the cases, 80% of the controls selected at random, apply the same classifier construction method to it, and somehow integrate all of the classifiers that come out of that. And that's pretty much, from my view anyway, that's pretty much the classifier that one settles on. And its AUC applied back to all the data, the 100% of the cases, 100% of the controls, its AUC should once again be pretty good, maybe nearly as good as the first classifier generated from all the data. So all this work should yield a classifier that is not driven by coincidences of a few markers with radical case control differences. Real biological data, of course, is fraught with difficulties. There can be missing samples or samples that are with readings that are ridiculously high. Different markers from the same experiment can have they have to be normalized. They have to be, the actual numerical values can be 1,000 or 1,000,000 times greater than each other, and so on. All those things have to be dealt with. But the method that I've outlined here, I think, will lead to a sound conceptualization of a classifier for the data. Now, there is still to do. Well, find an external test set, external data, and apply the same classifier. Ideally, the best practice is that the external data come from another lab by completely different persons, but using, of course, the same definitions of case and control, and that another analyst applies the same classifier and gets the same level of performance in distinguishing case from control. That's the ultimate, the external data. Now, this is a flowchart, which is too complex to go through, but this just simply, this is just a representation of the stringent random labeling test. So this shows the steps where we go through 1,000 and so on, and finally settle on the classifier that performs the best. And there again, this is a recapitulation of the implications, that the raw data had to contain information in the first place, that the normalization did not destroy the data, the information, and whatever classifier construction method was used discovered some of the information. And then there's the integration of multiple 80% classifiers to make sure that there are no secrets hidden in your data, a few samples that somehow drive false conclusions. And that's the pretty much wraps up the best practices. Now, I'd like to review in a remaining moments a couple of the shortcuts, and this is one that has appeared in the literature. It's an alternative random labeling test where the true classifier is applied to random data, and it should yield a null result. That is all completely true. It shouldn't yield a null result. And in this flowchart, the classifier is constructed and selected, and then the same true classifier is applied to random data. Now, of course, that's logically not the same as applying the true classifier to the true data and comparing that to all the random classifiers to the random data. However, this is a sanity check, and there's nothing wrong with doing it, but it's not the same as stringent random label testing. Now, instead of true classifier applied to random data, we can also try random classifier applied to true data. And that just means, well, there's a random classifier just means that the markers used in the true classifier are reselected, and so we go back to the original field of markers. I mean, we might start with 100 markers and settle on 10 that go into our finished classifier. We'll go back and just choose 10 at random from the 100 in the same structure of the classifier, and try that with true data. And of course, that should also yield a null result. And this is another method, a shortcut, one might say, that appears in the literature. There's nothing wrong with doing it, but it's definitely a shortcut, and it's not the same as stringent testing. And finally, another one is in lieu of external test data, some folks simply partition their data 50-50 or something like that. They set aside half the data and build a classifier by whatever means with half the data, and then they keep refining it until they're happy with the outcome, and then go back to the second half of the data and apply it. And once again, this is it should give a good AUC on the second half of all the data. But I would say this is again a sanity check. It's not very convincing. It could be that all those hidden relationships among the markers are actually what's showing up, and it could be that you're somewhere in the middle of that curve I showed you, where the AUC is good, but it's good for no really scientific reason. It's good by chance because of all the hidden relationships. So much better would be to do this 1,000 times, which is pretty close to the concept of five-fold cross-validation, where instead of 50-50, it's 80-20. So, regarding, and one last comment here on pathway analysis. Sometimes at the end of the day, after the classifier has been built, the modeler seeks to go back to the biology and support the conclusions of the classifier in terms of the biology, which is of course a great idea. And if there is biological coincidences or biological alignments, would be a better way to say it. If there are biological alignments, that's wonderful, and then that lends credence to the entire process. And by the way, back on that microRNA work that I showed at the very early on, the non-parametric study, the sequences, the A, U, G, C's in the microRNAs, they did, after all that analysis, they did have biological significance. And for those that are familiar with some microRNA biology, it turned out that the microRNAs that prevailed in the cytoplasm were predicted to target ALU repeat double-stranded RNAs, which in itself is pretty interesting because there are lots of ALU repeats in our genomes, and presumably they're generating a lot of double-stranded RNAs, and we want to get rid of them in the cytoplasm. So all that, that fits. That's nice. That's good. Okay, so in general, though, people will go to pathway analysis and submit their 10 selected markers that go into their classifier in which they now have much faith, and say, well, okay, what's the biological story? Well, if this goes into an engine such as Ingenuity, well, I think it's very likely the 10 out of the 100, or my experience anyway, is that it's very likely that the 10 out of 100 will lead to some low p-value pathways identified by Ingenuity or other reputable and complicated pathway engines. The problem is the researcher sometimes doesn't go on to do the following. The researcher should take of the field of 100 markers from which 10 were selected, the researcher should go back and choose 10 at random and plug it back into the pathway analysis engine. And the researcher should do this many times to be sure that this pathway engine outcome is not just a chance finding. And my experience is that ingenuity can find a seemingly convincing alignment with pathways a lot of the time, much more than one would naively, or I would naively expect by chance. So there is a danger there that this confirmation with pathway analysis, it can be misleading. And just like everything else, there is a need to go back and do random testing to make sure that there's not some hidden bias or assertion in the original selection of the 100 markers. If the true selection is superior in some way to most of the pseudo selections, then the pathway analysis can then be regarded as evidence of classifier significance. Okay, so these are, as you can see, many of my own opinions are mixed in here, and I'm sure that those listening will have different opinions in some cases. But the use of sanity checks is fine, but they are in themselves, in my opinion, they're not as significant and as convincing as stringent random relabeling. And stringent random relabeling means starting over from the very beginning with raw data and relabeling the cases and controls and treating that new data set exactly in the same way as the true raw data were treated. Okay, well, I think that's it for me, and I think now I go to screen share and stop, and we have an opportunity to continue with questions.
Very good, and I'll hop in here and just encourage anyone to click on the chat or the question tab, Q&A, on the left-hand side of your screen. If you just go over there, it should pop out, and you can type in your question. We'll read them to Dr. Jeffries, or he'll read them himself, and we'll have we've got about 20 minutes for a Q&A today. So please dive right in, folks.
I see there is a question from Dr. Zhu, so I am happy to entertain that question if I can.
It just says hello, Dr. Jeffries. It does not. Oh. There's no question in there.
Okay, well, please add the question if you wish.
All right, it might. I can hear people typing furiously. So it'll take. It usually takes a couple of minutes to do it, and I forgot to encourage people. Oh, here we go. He's from Thomson Reuters.
Very good.
Are you seeing the Q&A, Dr. Jeffries?
Yes, I am.
Great.
He's from Thomson Reuters. I see that. That's great.
In the meantime, as we wait, is there anything you'd like to add on to your talk, or if you'd like to, any reminiscences?
Well, I believe that a person who becomes acquainted with these issues of use of permutation testing or random labeling can be quite useful to his organization, because there was, for example, just a few days ago, an important paper came out on schizophrenia. And in the paper, the authors suggested they used random labeling, but I dug into one of the supplements, and in my opinion, they did not use stringent random labeling. And so quite a few other persons have come forward. Many geneticists have come forward on this particular paper and pointed out the same thing. So I feel that this is a body of knowledge which could be very beneficial to many organizations and many developers of biomarkers and so on. Anyway, I read now a question here. It says, if random tests get 65% accuracy and real test is 80%, how confident is the real 80%? Well, I would say that of course, the random tests, there's more than one random test, and they don't all get exactly 65% accuracy. So what counts is the distribution of their accuracies. And so if in this distribution, as in the graph I showed, if you have a nice histogram with many values, say less than 75%, pardon me, and only the one from that 80%, well, that would be very good news. And you can use that distribution, a beta distribution, a gamma distribution, or whatever, to calculate a p-value. And that is, if the p-value is very small, then of course that's goodness. That means you were successful. Okay, and so I'd see there's which metric is the best for assessing classifier's performance in the most case? Well, the area under curve of the receiver operating characteristic is considered by many to be a powerful indicator of the value of a classifier. Another one would be it depends on what you want to do, really. But it could be your metric might be defined in some other way in terms of specificity and sensitivity, or the metric could be defined in terms of the student t-test p-value, for example. The student t-test p-value calculation requires looking at a fraction, and in the numerator of the fraction is the difference, the absolute value of the difference in the two sets of numbers, and the denominator is made up of the standard deviation of the first set and the standard deviation of the second set. And so the greater, of course, the greater difference in the averages and the smaller the standard deviations, the larger that fraction, and the smaller the p-value. So that's another way you could say, well, I'm really separating my cases from controls very well if my student t-test p-value is small. There's nothing wrong with that either. But I think many would regard the area under curve of the ROC to be maybe the best way to put it is hard to beat. It's the best or near the best. Okay, so I guess there are other questions. How would you attempt to determine the number of classes represented in your sample if you assume you're not looking at a binary distribution? So how would you like to how can you estimate up front how many experiments you have to run to achieve significance? Well, the power calculations often assume normal distributions, and the fact is actually if you have 10 real numbers, they are normally distributed to some extent, not very much maybe, but there is a confidence level. And so this whole question of normality is a slippery one. And I would say go to your manager and say, well, I need to start off with 5 cases and 5 controls, and then you can draw your histogram, draw your distribution, and that histogram conceivably could already be so clear that there's no need to do any additional experiments. In other words, the 5 cases could be completely different from the 5 controls. On the other hand, if there's a lot of overlap, then you can say, well, I need to try it with more experiments, and you will have to spend more money, and so on. Now, that's the best answer I can give you on that one. A lot of people demand upfront, well, how many we have to do, and I don't know that there's really a good answer for that in the real world. Quick question from Dr. Elliston. Considering individual values, how would you best identify and treat outlier values as you're developing your multi-gene classifier? I would say talk to the chemist or the biochemist, and of course, if you in the case of blood analytes, if there's some substance which is ridiculously high, then that has to be treated as being well, that makes no sense. In RNA-seq and RNA sequencing, sometimes the runs fail, and so you have very few reads. And so if all the experiments have something like a grand total of, if all your samples have a grand total of say 100,000 reads of microRNAs, then and one of your sample only has 100, well, then chances are that assay failed. Also, and this is another very hard question, but I would say my practice is if you have to set an arbitrary threshold for discarding outlier values, that's fine. But do the whole thing over again with another arbitrary threshold. Now, in the best of all worlds, resetting that arbitrary threshold will not alter your conclusion. That's a very happy situation. On the other hand, if resetting that arbitrary threshold alters your conclusion greatly, or the quality of your classifier greatly, well, you have to tell your manager, I don't know what to do with these outlier values. I don't think there is any simple universal answer to that question, unfortunately. And I guess that's it for now for the questions.
It appears so. If there are any other questions out there, please feel free. We'll give a couple minutes for people to consider it. I know we have about 40 people. It looks like something just popped up again. Can you see that, Dr. Jeffries?
Yes, I do. It says if random testing results in greater than 75%, does this mean that the random test is not a truly random, but leaking some information during random tests? Well, as I showed in my the histogram in a few minutes ago, I don't think that an AUC of 0.75 is the end of the work. I think that it can happen in real data by chance. And so once again, it's just the onus is on the modeler to rerun those data many times with random case control labels and see what happens. Build a histogram. The tool that I like is called EasyFit. I have no financial tie with that company whatsoever, but I think they build a good product and they have more than 50 different types of distributions. Now, before I bought that product, I didn't know that there were more than 50. Aside from normal, gamma, log normal, beta, and so on, there are many other ones. And that kind of if you can make a histogram with 1,000 numbers, then and fit one of those distributions to it, then I think you can feel a lot more comfortable about coming to a conclusion. Okay.
All right, very good.
How would you determine the number of, here's a number of classes represented in your sample if you assume you're not looking at a binary distribution? That is a very important and interesting question, and I do not know the answer. Of course, clustering algorithms will cluster your data for you. The trouble is, and this often is not mentioned in the literature, there are many clustering algorithms, and I can take the same matrix of data and plug it into 10 different clustering algorithms, and sometimes you'll get exactly the same clusters, which is wonderful. Sometimes the clusters will be completely different, which is not wonderful. So this is an unsolved problem, in my opinion. I guess that's all the questions from.
It looks like it. Let's give it another 2 minutes. I think there are probably not all of us are even capable of asking questions about this. It's a fascinating subject, but it's pretty complex. So we'll give it just another minute or so. Folks, if you have any other questions, please type them in.
I will say that this random labeling in drug development, and I have colleagues in drug development who actually have introduced me to this whole topic. They call it Y randomization, the letter Y randomization. And I will also say that it's by no means original with me. random labeling goes back a long way, and in fact, a statistician friend told me it goes back to R.A. Fisher himself, which would mean about 80 years ago. But in those days, there were no computers. But Fisher apparently, or Sir Ronald apparently, surmised or suggested that it would be great to, this would be the sine qua non, this would be the ultimate test of a classifier, is to mix up cases and controls at the very beginning and reapply the entire protocol exactly the same way, and then one would truly know. And so if he's looking down on this now, I hope I'm not making too many mistakes.
We have a new question from Harry Athanasiou, or Athanasiou, pardon me for mispronouncing.
Oh, all right. People always say the sample size is too small comparing to the number of genes when using microarray gene expression data. Could you suggest what is the safe number of samples for classification analysis for such data? Well, again, if you were really fortunate, you could have 3 cases and 3 controls, and if the differences were dramatic enough, then you could call it a day. Or you could use this non-parametric analysis that I went through and say that, well, not all of the markers are necessarily informative, but there must be some in a certain set that are, as was in my 3 versus 3 discussion a few minutes ago. But in general, microarray data, it doesn't work that way. And you could try non-parametric analysis looking at ranks. And if that doesn't work, then your only recourse, in my opinion, is to, as I said before, try it with 5 versus 5, 10 versus 10, 20 versus 20, and keep building your histograms all along the way and fitting the distribution. And at a certain point, you can say, well, now I've got enough samples, and with this fit of this distribution, my p-value is so small that I can go to management and say, yep, this is how to differentiate case and control. Or you can say, I tried it with more and more samples, and I never got a good differentiation, and so I don't think it can be done. So that's the best I can say on that one, okay?
Very good. And there's a question right above that that begins with the addition of surrogate latent variables. Do you see that one?
Yes. With the addition of surrogate or latent variables, analysis to remove unwanted variation for the gene, the classifier can seem a lot better than it is. Well, I think in drug development anyway, when there's a lot of money on the line, anytime there is an arbitrary introduction of variables or an arbitrary setting of a threshold, it's a very good idea to spend the hours and keep resetting those decisions and to discover whether or not those decisions truly impacted the conclusion or the quality of the classifier. So I think that's the best I can say on that one.
All right. Well, last call for questions. We've got just about 5 minutes left in our session. We'll give it another 90 seconds here. There's some really great questions. And you are coming to us live from Chapel Hill, North Carolina. Is that correct?
Yes, that's completely correct.
I looked up Chapel Hill yesterday, and it's one of those unique, not that unique for a college town, but the number of students and employees of the university and medical center exceed the population of the town by about 40%, 35%.
Yes, that is the truth. So riding on the bus every day, I pretty much am in the company of a lot of researchers and medical doctors, and as well as students. That's true. It is for the most part, a wonderful population and a great place to work.
Looks like we might have one last question here. What is the best way to deal with extremely unbalanced classes, and which metric is safe for assessing the performance of classifiers for unbalanced classification analysis? Is that that's a new question, I believe?
Okay, by unbalanced, that's many cases versus a few controls, or vice versa, I would think. And I think there is a very good way to approach that if the ratio is not too extreme. And that is, suppose you have, let's say, 50 cases and 500 controls. Well, go into your cases and select 25 at random. Then go into the controls and select 25 at random. And do some analysis with those random subsets of 25 and 25. And now this is going to require some serious computer time, because if the number of controls is so great, to really get a proper or a survey of all those controls, it's going to take a lot of simulations. And maybe I'm exaggerating these numbers too much. But I think that is a good way to, is taking random subsets of equal sizes of the two classes or groups. That's a good way to approach it. And I've been doing some work on this where I looked at correlations of markers within cases and correlations of the same markers within controls. And Pearson correlation depends a lot on the definition. I'm sorry, on the dimension. The higher the dimension, the less likely it is that a given threshold will be exceeded by Pearson correlation. So a way to approach that is to look at subsets of the same size, but do it many times. So that can offer some help in that topic, I think. Okay?
Very good. And seeing no more questions, and this being just a couple minutes ahead of the hour, we will wrap it up. I will first thank Dr. Clark Jeffries for delivering the presentation, and also pitch our next webinar, which is coming up actually in just 3 weeks. It's so thank you, Dr. Jeffries, and thanks to everyone who tuned in and participated and asked the great questions. Our next webinar is entitled Brain Code, a neuroinformatics platform for brain discovery, presented by Francis Jeanson, the program lead informatics of the Ontario Brain Institute. That will be on Thursday, October 23rd at noon EDT or 9 a.m. PDT. If indeed daylight savings time is still, I can't remember if it's on or off at then, but anyway, noon and 9 on the 23rd. We'll be sending out reminders of that. Once again, thank you, Dr. Jeffries. Thanks everyone for tuning in, and we will see you next time.